Hello.
Hi.
Hi, how are you? So how's any research updates or anything like that?
I think the dean signed the form to say that I could be on their digital system, so I should have full access to libraries again
Oh, good.
and get on with things like write a paper. Um, yeah, I had to outline what I was planning to do, so it's like write my paper about my thesis, and then do animal care course so that I can work with the axolotl eggs. Then I have to learn how to run the optical coherence tomography equipment, and then I can take images of eggs.
It's a lot of leadway going into it.
Yeah, so he said, I'm really busy this fall. How can I do this fall? And I'm going, well, I have to do all these other things. So no, it's not until spring. And so he liked that.
Good.
When he's teaching, he doesn't want to do any extra projects.
Right.
Yeah, I understand that.
Right.
Because I've taught. I understand.
Yeah.
Yeah. Anyway, so yeah, it looks like I might be able to do that. It would be nice to get a baseline images of the developing blastocele.
Mm-hmm.
Yeah.
Yeah, it'd be great.
Nobody's done it. I keep
Really?
telling them that nobody's doing this research. So at least not. I could do a search, but the number one they use Xenopus, and number two they're not doing that.
Huh. Okay.
This is the engineers and the biologists don't talk to each other.
Right.
Because there's a perfectly fine experiment done on Xenopus. Anyways, it's with optical coherence tomography.
Right.
They looked at the movement of the layers inside the egg. So I just need to duplicate that with the salamanders.
Well, that's great. Hopefully that works out well and you get some good results.
Yeah, well, at least, and with animal care, if somebody's done it before, they're more likely to say, "Oh yeah, this has been done and on a slightly different species, and it's good to compare species." And then if I get a good paper that I can send it to the herpetologists.
Oh yeah.
And maybe they'd like to do that with some of their what is it, blue-tailed skinks.
Oh,
They grow in Manitoba. Blue-tailed skink eggs.
oh.
Anyway.
Yeah. All right. So that's great. Yeah, thanks for the update. So I went back after last week's meeting. I went back and looked for CompuCell3D methodology because last week we talked about ComCell, and I found a paper where they were doing some things with embryos, I believe, or embryogenesis, and I'm going to go through that, and I'm going to go through the methods, and we can compare those with what we saw in ComCell, which was, of course, implementing a mathematical model in a platform, and then CompuCell3D, of course, does that differently. So
okay, it looked similar to CompuCell3D though.
yeah. I mean, we'll see what they do differently. I just wanted to put them side by side and see. And this is a different system, but it's I tried to pick. Well, they've done some stuff in development in CompuCell3D. They've done chick embryos in this paper here, and then there are some more things on tissue regeneration, which is of course different, but it has the same sort of things that you need to model. So,
Oh, okay.
yeah.
I'm still interested in stress and strain curves.
Yeah.
Or force and change in distance
Okay.
curves.
Yeah. Yeah. Well, we'll see. I don't think they have any in this paper either, but you know, so it looks like we have someone here, Akshay.
Hello. Hi.
Good morning.
Good afternoon. Sorry, I just saw. So I was just looking for webinars that I could learn from my master's student from stem cell biology in India.
Okay.
So just for this webinars online. I'm sorry. I could have access to some weekly seminar kind of thing. Is it a workplace computer from some questions different amount?
Okay.
Oh, well, that sounds like he's interested in things.
Yeah. Yeah. I don't know. Gonna reconnect here in a minute.
Okay, and I'm probably going to chase a fly or two.
Okay.
They're bad this time of year. I'll wait for him.
Yeah.
Yeah, I asked someone who knew about ComSol, and they said, well, you have to make your truss structures large enough to get a good mesh. And I'm going, well, they're in micrometer size, maybe kind of nanometer size.
Yeah.
So how am I supposed to get a mesh? Because they don't do, they do nanotech in the solid mechanics, but they don't do it for trusses.
Right. Yeah.
And the trusses are supposed to be self-meshed. Like there's, anyway.
Yeah.
I don't know. There's a lack of information, I'm just saying.
Yeah. Well, yeah. Why don't we get started with the paper? So this is the paper here. This is called A Mechanical Model of Early Somite Segmentation. So this is from 2021. So it's fairly recent. And yeah. And then, so the graphical abstract, they show what's going on here. So this is unsegmented epithelium. This is the apical side. This is the basal side. There's this activation front, and there's this gradual increase in apical contractility up here, going along the activation front. Then this is the buildup of tension at cell junctions. So as the activation front continues down, the apical contractility causes this buildup of tension at cell junctions, as you see here. And then as the activation front continues on more, almost to the end of the sheet, then you start to get these separations at breaking tension. So you get these segments that form. So you get this apical contractility at one end. You get this buildup of tension at different junctions, and then they break open and separate at some breaking tension, and you get these segments. So that's the idea here. So their highlights say dorsal presemitic mesoderm of chicken embryos epithelializes before somite formation. Dorsal epithelium shows signs of apical constriction and early segmentation. A mechanical instability model can reproduce sequential segmentation. And then a single ratio describes spatial and temporal patterns of segmentation. That would be this here, where you get these separations and segments, and then this ratio, which I'll talk about in the paper, describes this. So this was published in iScience. So if we go into the summary, we start off. Somitogenesis is often described using the clock and wavefront model. So this is where you have the wavefront that we saw in the image with a clock, which is a timing mechanism. And this is, of course, a famous model in developmental biology to describe differentiation. And so that's usually how we describe somitogenesis specifically. However, this does not explain how molecular signaling rearranges the presemitic mesoderm, PSM cells, into somites. So these presemitic mesoderm cells we saw, which were ahead of the wavefront, are then, being rearranged into these somites. Our scanning electron microscopy analysis of chicken embryos reveals a caudally progressing epithelialization front in the dorsal PSM that precedes somite formation. So this is again this it goes caudally and it's moving. It's this front that of course we saw in the image. Signs of apical constriction and tissue segmentation appear in this layer in layer 3-4 somite lengths of caudal to the last formed somite. So this is where we have basically this localization of this apical constriction and tissue segmentation. We propose a mechanical instability model in which a steady increase of apical contractility leads to periodic failure of adhesion junctions within the dorsal PSM and positions the future intersomite boundaries. So we get this sort of, as the front goes along, we have these mechanical instabilities, and this leads to periodic failures, which give us these boundaries. This model produces spatially periodic segments whose size depends on the speed of the activation front of contraction, which is parameter F, and the buildup rate of contractility, which is this symbol here. This ratio, which are these two, the buildup rate of contractility over the front of contraction, determines whether this mechanism produces spatially and temporally regular or irregular segments, and whether segment size increases with the front speed. So we have all these things that we're trying to figure out about it. What was that?
There's also a physical phenomenon that separates things into drops, like a long line of honey separates into drops. So there's also a surface tension phenomenon that helps with the somite formation, as well as perhaps this is another wave, for instance, that Dick keeps talking about. It's a wave of contraction that goes down the precursor spine to form the somites. But there is that phenomenon that breaks apart a line of I don't know. Honey is fairly dense liquid. I should look that up. I'm forgetting what it's called, so I'd have to look it up. But so there's a surface tension phenomenon also.
Okay. Yeah.
It's in play.
Okay. Yeah, exemplified by honey. It's like Morgan's here. Hello, Morgan.
Good morning.
Yeah, good morning. So we start off with talking about somitogenesis. Somitogenesis in vertebrate development sequentially and periodically creates metameric epithelial balls, which are these somites, along the elongating embryo body from the bilateral rods of loosely connected mesenchymal cells called presemitic mesoderm. So you see that sheet of epithelial cells, how they're being kind of pushed apart by these forces, and then they form these sort of subsets of epithelial cells that then form balls. And like Susan said, this resembles drops of honey, where basically it results in these balls, and so they kind of beat up on this surface. As cells leave the rostral anterior head, end of the PSM to form each somite. So rostral anterior is where you have head to tail, anterior being the head, rostral being the tail. And this is just kind of but this is the head end. So you have this end here. To form each somite, new cells continuously move from the tail bud to join the PSM at the caudal posterior or tail. So you have the rostral anterior, which is the head, the caudal posterior, which is the tail. That's the sort of the terminology. The anterior posterior is what we're used to in C. elegans. Rostral caudal is what people like to talk about, in anatomy as well, and it just depends on the convention people use. But that's basically it. We're going axially, and this front is moving, and we're seeing this phenomenon unfold in that way. Okay. At any given rostral caudal position, a pair of nearly equal sized somites form simultaneously on both sides of the neural tube between the ectoderm and the endoderm. So this is where we have these nearly equal sized somites, and we have this pair happening at each position or in any given position. So the position is, I guess, random, but you get these equal sized somites forming at that position. And then this is on both sides of the neural tube between the ectoderm and the endoderm. These transient structures are the precursors of vertebra, ribs, and many skeletal muscles. Many birth defects arise from a failure in one or more of these developmental steps. So you get, if you have these sort of somites, they sort of form these different components where you have, they're like in vertebrae, you have, of course, different types of vertebra in the vertebral column. You have different numbers of them. And so you have this sequence of things moving down the body axially. So this is important to get this sort of mechanism in place where you can make these kind of repetitive depositions, as it were, of some thing that, some structure and development. Okay, so, if we don't get this to work right, we get birth defects, and they've listed them in this reference here. Oh yeah, Rayleigh plateau instability.
Yeah, I have in something I wrote, the physics is Rayleigh plateau instability, and it was also called capillary thinning. So anyway, I have a paper on it.
Yeah, okay. Oh, you wrote it, or did someone else write it?
I wrote it, yeah.
Okay.
As a part of something that I needed to do for, it was a final essay for a course. And I prefer to write a small exam and then a big essay. I'm not into exams.
Okay, yeah.
Anyways, would you like to see my screen, or?
Yeah, let me do this. You can share your screen. Well, you can share your screen, yeah. And
Okay, share screen. I think it's that. Okay, that's not working. Okay, window. Oh, there it is.
here we go.
And this is a zebrafish developing, and these are some examples of this type of phenomenon, and they tend to be periodic.
Okay.
Fairly regular periodicity. And I have several references here. I don't know if I can pronounce that. Snowger et al. 2020.
Yeah. Yeah.
Anyways, I did this with some honey one time, and Kamran Nuri used it in his publication. And I just put a thin line of honey on a piece of wax paper. Dick instructed me to do this, and it did change into a periodic structure like the bottom part here.
Okay.
So there definitely is this type of instability that is naturally occurring.
Okay. Yeah. Yeah.
Just wanted to point it out because, yeah.
Okay. Yeah, that's great. Thanks. Okay. So let's see. And so if we look at like somitogenesis in terms of being robust to perturbation, it's strikingly robust to perturbation, both spatial and temporal. Changes in the total number of embryonic cells and the rate of new cell addition at the caudal end of the PSM lead to compensating changes in the size and timing of somite formation so that the embryo eventually produces the same final number of somites as in normal development. So there's this sort of compensation when things don't go the way they're supposed to, or at least to an extent, because you can get developmental defects. A linear increase or scaling of the somite size at the speed of the caudal moving position of the determination front, or with the rate at which cells join the caudal end of the PSM, can result in this number conservation. So this is, again, a number of internal mechanisms working to regulate this. Models seeking to explain somite formation include the cell cycle model, which couples the timing of segmentation to the progression of cells through the cell cycle. So this is where, every time there's a cell division, you have this timing mechanism, and it corresponds to the timing of segmentation, along with the cell intrinsic gating mechanism. There are also reaction diffusion models, which can be used for pattern formation. And so currently the most widely accepted family of models employ a clock and wavefront mechanism, which we talked about, which combines caudally progressing fronts of determination and differentiation with an intracellular oscillator, which determines cell fate based on its phase at the moment of determination. Following the identification of the first oscillating transcripts, which are Hairy 1 and Hairy 2 in the PSM, so this is the upregulation of oscillating transcripts or transcription factors, and then many computer simulations of varying complexity have implemented different CW models. So the clock and wavefront is driven by these two transcription factors, and you can model it at that level and get a timing mechanism and all that. Most CW models reproduce the experimentally observed scaling of somite size with clock period, front speed, and rate of elongation of the PSM. Okay. Recent experiments have shown that somite-like structures can form without either a clock or a progressing determination front. So you can get these sort of structures sort of without these mechanisms, and you can sort of get a model-free account of this sort of pattern formation.
Yeah, maybe that's what I just showed you.
Perhaps. This is Cottrell 2015 and Diaz 2014. So Cottrell 2015 is a local self-organizing reaction diffusion model, can explain somite patterning in embryos. This is using explicitly a reaction diffusion model, but as a local self-organizing type. So I'm not sure, I'm not familiar with that, but that would be the reference there. I was in Cell Systems. And then the Diaz reference, this is with James Glazier on it, somites without a clock. And so this is, from 2014. I'm going to assume this is some sort of like segue into the CompuCell3D.
Yes.
Yeah. The ability of somites to form without either a clock or a front suggests that we should consider other mechanisms that could lead spatially and temporally periodic sequential division of the PSM into regular segments. Recent experiments by Nellemans showed that applied tension along the rostral-caudal axis can induce the formation of intersomatic boundaries in locations not specified by CW signaling. This suggests that mechanical mechanisms may be important in generating intersomatic boundaries. That's the mechanical mechanisms we saw in the diagram. Mechanical instabilities could also result from invertebrate segmentation, assuming relaxation of junctional adhesion sites as the PSM elongates. Their pre-patterning mechanism generates a number of somites independent of the final segmentation mechanism. Their model, however, does not consider the sequential development of the boundaries nor any active processes within the tissue. So they're trying to work out a sort of a, I wouldn't say it's a model-free model, but they're basically trying to work, use CompuCell3D to investigate this type of organization in more detail. So our observations showed that the dorsal PSM cells undergo early maturation, forming an epithelial monolayer along the ectoderm boundary, beginning long before somite formation. The presemitic epithelium also shows signs of presegmentation, with clusters of cells forming arched tissue segments, roughly the length of a somite diameter. Those were those bundles that you saw in the graphical abstract. These observations suggest that periodic tissue segmentation and somite boundary positioning could result from mechanical instabilities, similar to periodic cracking of materials subject to stress.
Mm-hmm.
Yeah.
Yeah.
We developed a model of the dorsal epithelial monolayer where the observed boundaries between dorsal segments arise from the loss of contact between two neighboring cells due to increased apical tension between cells. We simulated this scenario with a 2D computational model of a cross-section of the epithelial monolayer and showed that a simple mechanical model without a clock can explain the spatial periodicity and segment sizes. We also showed that this model can produce either roughly constant size segments or segments' size increases or scales with activation front speed and inverse rate of increase of apical contractility. A critical threshold for the ratio buildup, this rate of apical contractility to the activation front speed, defines the boundary between these two domains. So this is this ratio that we're talking about. A second threshold for this ratio predicts whether this mechanism produces spatial and temporal irregular segments or irregular segments. So you have regular segments or irregular segments, basically. And so this threshold for this ratio predicts that. Okay, so I'm going to go down to, so this is kind of maybe a decent sort of set of images to show this process in vivo. So this is early signs of presemitic epithelial segmentation. This is electron microscopy showing this process, the dorsal here, the ventral, the rostral, the caudal. And so you can see this sort of process happening. This is 100 microns. So you can see this process happening kind of from caudal to rostral and how this is working. So they show these somites being 100 microns, about the size of this scale bar. And then they show the PSM here, S0 and S1. They show these two somites and then the presemitic cells here. So actually it's rostral to caudal is where things are going. So this is somite already formed, and then the rest of these cells down here, these epithelial cells, are not organized in that way yet. So you see that the organization is going caudally. Okay, so then they just show the aspect ratio here. These are the the organized epithelial cells. So you can see that. You can see the apical end there. And then you can see this graph, which is where you have cell pair, and so different size cell pairs from rostral to caudal, and then you have side-to-side distance in microns, and you see that you have these, this is S1 over S0, S2 over S1, S3 over S2, and you see how at the apical end and the basal end, you get these different side-to-side distances. There's a difference between them. As we go from rostral to caudal, the difference, I guess, gets smaller. I'm not sure exactly how the trend is shaping out, but basically it replicates what we saw in the graphical abstract. So that's kind of something to kind of prove that this is kind of quantifiable, and we can see this in nature, in vivo. Okay, so we have this mechanical model, and they kind of talk about some of the things here. This is the proposed models of segment formation. So these are different. So A here is the epithelialized model, or epithelialized cells. So this is kind of the initial state. This is the model's initial condition, where everything is stable. You have the basal side, the apical side. You have rostral to caudal organization. And then this set of models here in B is this mechanical instability model. This is what we saw in the abstract, where you have the activation front moving down caudally. You have the gradual increase in apical contractility here. You have this buildup of tension at cell junctions, and then the separation at breaking tension at different points. Then you have this cell clustering model, which is where you have the activation front again, but this time instead of having this apical contractility, we have these random cell-cell associations. So we have apical contractility happening, but they're pairwise instead of gradually down the sheet. And so you get these cell-cell associations, you get these groups of epithelial cells, and then they start to have these separation points. So you get these clustering, you get these separation points, you get clustering into larger cohorts, and then you get the separation point here, which gives you a segment. Okay, so that's the cell clustering model. And then the seeded activation is where you have different seeds where the cell is activated. It recruits other cells through signaling. You have another seed which does the same thing, and that kind of forms the segments from within, because the segments are only as long as the cells that are recruited by the seeded cell, the focal cell, and in between where those influences fall off, if you think about it like gradients in a reaction diffusion model, you get separation where those gradients sort of intersect. And so that's how you get your somite. So they have these three models that they have proposed. And so then they say our mechanical instability model has three main parameters: F, this symbol, and F break, or this break parameter. So we have the tissue, which is F, the rate, and that's what we refer to as the buildup rate of contractility. And then let's see. And there's a maximum tension, the length between adjacent apical domains of neighboring cells can sustain before the cell loses their connection with each other, and that's break. So it has this sort of the tissue itself, the rate of buildup of contractility, and then this breaking threshold. Okay, so this leads us to the Cellular Potts model, and of course CompuCell3D. So we implemented a stochastic cellular Potts, Glazier, Graner, Hough model, which is the sort of the basis for CompuCell3D. This model version of dorsal tissue segmentation. So this has been talked about in these different references here. In this model, the cells are spatially extended objects similar to the schematic cells in figures 2A through 2D, with the width and height, apical, basal, and core domains, and elastic connections between neighboring apical domains representing apical junctional adhesion domains. We first characterize the model behavior for different fixed values, and then for simultaneous increase of that fixed value in all cells at this rate here, before exploring the effect of a gradual, caudally progressing front F of apical constriction activation. So the front of in the tissue is F. This is just the constant that's going through the tissue. Okay, so then we first tested our model by creating small epithelial monolayers, fixed aspect ratio. We increased the strength of apical contractility of all cell pairs simultaneously at a fixed buildup rate to a maximum value ranging from 20 to 600 without allowing apical links to break. We then observed the shape of the tissue and the average cell pair tension over 20,000 simulation time units. This is done through Monte Carlo. And then after our parameter reached its maximum value, as the number of cells in the monolayer increases, cell pairs in the middle of the segment experience higher tension than cell pairs near the periphery, which maybe is expected because in the periphery it doesn't have as much buildup. This dependence on tension and position within a segment forms the premise of our model. And that is, as the tissue becomes larger, tension between cell pairs increases. Since the junctional bonds between cells have a defined breaking tension, higher local tensions predispose the tissue to break into smaller segments. So this is kind of the setup here. This is A is the snapshot of 4 simulations of 10 cells. So we have these 10 cells here with different levels of maximum apical contractility strength. The tissue becomes more constricted for larger values. And then this is 20. This is 100. This is 300. And this is 600. And as you can see, it's kind of like this part here, apical constriction is happening down here. So this is kind of being pulled inward. And you see it's almost like it's like a bowl by the time you get down to 300 and 600. Whereas in 20, it kind of looks like still looks like a long segment of cells. And you get, some limited apical constriction, but not a lot. And then, so we have B, which is a plot of the average apical tension between cell pairs at the end of multiple simulations with different numbers of cells. So this is where we have our cell pairs here, which goes up to 24. And then we have our tension, which is in negative values going down, so from negative 2,000 and negative 6,000. And you can see this where you have, for cell pairs, it goes down maybe to, in most of their examples here, the lowest tension is found at about 12, or between 10 and 15, and then it goes back up. So you're thinking about how many cell pairs are exhibiting the maximum tension, and it's basically this curve where if there are too many cell pairs, the tension decreases again. Then this graph C, maximum tension and the number of cells. That is the maximum cell pair tension versus the number of cells in the tissue for different maximal values. So you can see this here. And this is for these different parameter values that we see in A. So as we go down to like 600 and 800, we get this maximum tension is decreasing, and it's as much as any other example. So the maximum tension is, going into larger negative values as we go down to a larger number of cells. And then for 50, it doesn't really matter because the whole thing, it's basically zero maximum tension. For D, in D&E, we have a histogram of distribution of segment sizes and normalized segment sizes for different rates of simultaneous buildup of apical contractility. So this is where we have the buildup rate. I can zoom in a little bit here. We have the buildup rate, which is this parameter, and we have this for different parameter values. We can see that it kind of shifts as the parameter value decreases. It kind of shifts to the right. So then the segment size gets larger on average, I guess. And it's going from like this. Well, actually, it's not really a trend like that. It's kind of like 0.5 is here with a mean segment size of about 8. Then they think this is 0.1, which has a mean segment size of 4. And then 0.025, which is the blue, is a segment size, within, it's maybe bimodal around 14. So you can see that kind of, it's just showing the segment distribution frequency there for different values. And then for E, we have this broken out for these different parameter values. So we can see that this is normalized segment size. We can see the shape of those distributions there. Okay. So, we're learning a lot from this model. So this is not, like in the case of our model that we ran in ComCell last week, there was a lot of mathematical modeling. They were building an Eulerian, and then they were building a Lagrangian representation of space, and they had this mesh that they then simulated everything with. And they were showing this basic movement of cells, or the cell motion, this invagination. And here we're seeing this apical constriction and this formation of patterns. But we're doing it in a little bit different way. We're using these cellular Potts models, which basically model things at the level of cells, and then they exist in a matrix, and then or in an array, and then you can manipulate them and measure these different features. So the cells are interacting, so you have the ability to simulate interactions between cells, and it's a little bit different model. And so they kind of get into some of their results here. This is actually a nice figure 4 here, where we have this rostral to caudal progression. We have these different separations here that you see. So you actually get, and this is where, we saw, like I said earlier, that it was kind of random, but it's also regular at the end of the process. So this A is where you get this time series simulation showing the sequential segmentation of a tissue due to a linear increase in this parameter. The cells progressively activate rostral to caudal. So this is where we get this, these kind of divisions, and then they kind of form these clusters, and then the clusters sort of align themselves to get this regular pattern here. So this is over time, where you're getting this process down the line, and then it's forming these regular clusters, these regular somites. This just shows these graphs here, the tension versus time. So you get these different cell pairs, different colors. This is the first segment, this is the second segment, so you can compare the two. The first segment, you get this decrease in tension until you get to a certain point in time, and then there's this, I guess it goes down in terms of negative tension and then up. And then you get this, it's a sharp transition here. Actually, in this case, you have two sharp transitions. The first one that's labeled in red, and the second one's labeled in blue. And then in this second segment, you get the same thing. You get these two sharp transitions, but they happen a little bit later. And so and then this is the tension for the first segment and the second segment in 3 different conditions. The red one is at rostral tear, the blue one is at caudal tear, and then black is at both tears. So the tension varies.
Yeah, I'd like to compare that with Rayleigh plateau instability for the same material.
Yeah.
It'd be interesting.
Yeah.
I'm sure a living system changes the periodicity of the segmentation, but anyway.
Right. Yeah, it's going to be a lot. Well, yeah, I mean, in simulation, it's going to be a lot more idealized. So figure 5 is where we have segmentation as a function of activation front speed, where we have this buildup rate, we have segmentation size on the y-axis, and then front speed in terms of cells on the x-axis. And so we can see the front speed as the front speed increases. We get a general increase in segment size here. In A, this is average segment size as a function of front speed. And in B, we have average segmentation time, which decreases as a power law with this exponent here of the front. Each line shows a similar behavior for different base values of the buildup rate of apical contractility. So you see in the first one, you see this average segment size as a function of front speed, and then the second one, we have this power law scaling in terms of segmentation time as a function of front speed. Okay, so I did want to show this other paper which describes this kind of phenomenon. This is a different paper. This is way back from 1992. This is the Graner and Glazier paper. So this is in Physical Review Letters, and it's called Simulation of Biological Cell Sorting using a two-dimensional extended Potts model. So this is where we have this biological cell sorting. I mean, you can do this for all sorts of different phenomena. I mean, basically, pattern formation is about cell sorting. And so you can do this in this extended Potts model. And this describes sort of the physics behind this model. So this isn't just like, magic or boxes interacting. This is where we have some real physics behind this. So in this paper, we talk about simulating the sorting of a mixture of two types of biological cells using a modified version of the large Q-Potts model with differential adhesivity. We find long-distance cell movements leading to sorting with a logarithmic increase in the length scale of homogeneous clusters. Sorted clusters then round, which is a complete sentence that was published in Physical Review Letters. Not sure what that means, but we find two successive phases: a rapid boundary different creation of low cohesivity cell monolayer around the aggregate, and a slower boundary independent internal rearrangement. So in this simulation, they're kind of sorting things, and they're forming this round shape, and there are these two phases where you get this cell monolayer around the aggregate, and then a boundary independent internal rearrangement. And so what they're using here is they're using Hamiltonian. Experiments have demonstrated the differences in intercellular adhesivity determine the final state of cell sorting, the cell configuration approaching the global minimum of overall surface energy. Driving forces can arise from differential surface energies. The differences of surface energy for boundaries between like cell types, homotypic, unlike cell types, heterotypic, and cells in the external medium or edge. So previous simulations of cell sorting on a lattice, so this was pre-1992, and using other methodologies, either took cells to be point-like or used non-realistic rules to describe topological changes. So they're interested in topology of the cells. However, cells have a non-trivial topology, which must be simulated correctly if we wish to understand cell sorting. So we need to understand the topology of this sorting process in order to understand cell sorting.
There's an interesting paper that in microgravity, cells sort according to size.
Yeah.
Because of, I guess, different surface tension, stronger surface tension if it's a small cell. I don't know. But yeah, they tend to clump according to the cell size.
Yeah.
And can disrupt an embryo.
In this respect, boundary and vertex dynamics models are preferable. The large Q-Potts model is proved useful in simulations of diffusive grain growth driven by surface energy. It can correctly simulate experimentally observed topological changes in cellular patterns in metals and soap froths. So you can do this sort of thing where we have soap bubbles in a froth, or we have different patterns in metals, and we can describe those using the large Q-Potts model. Therefore seems a good starting point for a cell sorting model. However, cell sorting occurs through the movement of cells rather than through cell growth. In addition, the Potts model simulates a pure material with a single surface energy, while cell sorting requires the presence of at least two different cell types. So they're going to simulate cell sorting in two-dimensional aggregates using an extension of the two-dimensional Potts model, which constrains cell size and allows for different surface energies between different cell types. So their model is as follows. The large cube Potts model describes a collection of n cells defined by defining n degenerate spins, and so they have that as sort of a lattice site. A cell consists of all sites in the lattice with a corresponding spin. So the physical version of this is a spin glass model, and they're basically mapping this to a biological system. Cells need not to be simply connected. Mismatched bonds between different cells have energy 1, and bonds between like spins have energy 0. The Hamiltonian, then, if we write that down, is the Potts Hamiltonian, which is just basically the summation of neighboring cells, and then this term here, 1 minus this term, and I don't know what that term is, what it's referring to, but in any case, this is the Hamiltonian that we're using. So where the neighbors may be of any desired range on either a square or hexagonal lattice. At each step, we select the lattice site at random and change its spin with Monte Carlo probability for a temperature T greater than 0. And then we have, so we can have change in the Hamiltonian, which is the energy gain produced by some change. There is critical temperature Tc, above which each cell dissociates. Below Tc, the spin coalesces into compact cells, though for finite temperatures, isolated mismatched spins are possible. At T equals 0, the cells grow by relaxation as the pattern minimizes its total surface area. The pattern's length scale increases as cells disappear. The dynamics and statistics agree well with experiments on surface energy-driven grain growth, such as soap froth. So they have this model, they build it for soap froth, and it's possible to see this. Okay, so that's in a sort of a purely physical system. So in cell sorting, we have a slightly different problem. Well, the surface energy driving mechanism is the same as for grain growth. Biological cells have generally a fixed range of sizes. Thus, the pattern cannot lose energy by coarsening, since cells cannot disappear. I mean, they can die, but they can't disappear. And this is something that we see in bubbles in magnetic films. And so we're constraining the evolution of this cell population. But instead of that, we look at differences in contact energies between cells of different types, which we characterize as differential adhesion. And this causes cell motion, which reduces the pattern's energy. To include these ideas, we add an elastic area constraint to our Hamiltonian and introduce a second quantum number, which is the cell type. So I think what we're getting is we're getting our spin, and that's corresponding to cell type or some characteristic of the cell. In our simulations, there are 3 cell types, light, dark, and medium, and that's included in this term tau. The surface energy between 2 cells then depends on the types of cells. So each cell still has a unique spin and consists of all lattice sites with that of spin, but there may be many cells of each type with the same tau. There is no simple way to introduce the differential surface energies without the cell type. So then we write down the modified Potts model here, and this just shows the spin types, the neighboring cells, and that's an additive term. Put that together into the sorting Hamiltonian. And so then we can actually model this process. We then use Potts dynamics with 1 Monte Carlo time step, defined to be 16 times the number of spins in the array, but we suppress the nucleation of heterogeneous spins by requiring that a lattice site flip only to spin belonging to one of its neighbors. The constraint is biologically realistic for compact aggregates, that we could relax it to allow the nucleation of medium-filled vacancies. We have checked that for our initial conditions, relaxing this constraint makes no difference at t equals 0, only a negligible difference for t greater than 0. So, if we have these, um, different constraints, they make a difference or not. Okay, so then, they kind of walk through this model and they show kind of their results. And this is actually, they have a nice visualization for 1992, which is cell sorting. So this kind of shows how this model, the output of this model. And so, again, you have these cell types. They're light and dark and kind of a medium state. And you can see them being kind of randomly distributed in these examples here at the top. And then in C and D, you start to get this, where the light and dark cells coalesce, or the medium cells coalesce into different groups. So you have this coalescing of the black cells into these sort of patches. And then you get more coalescing in D. And then finally in E, it becomes this blob within these other cells. And then finally in F, you get this sort of this, inner cell mass and these cells around it. So you get this sort of sorting into two circular compartments.
Yeah,
So that's how this.
I wonder how that fits in with the blastocell. Anyway,
I'm not sure. Yeah.
Or a spheroid, or.
Yeah, I'm not sure.
Yeah. Anyway, it's become quite a sophisticated model. Yeah. I took the course.
Yeah.
I could take it again. Like, it was fun.
Yeah.
That's why I was learning how to program in Python and learn the model at the same time. But yeah.
Yeah. Okay, so I think that's enough for that. And I think that's it kind of contrasts with the ComSol work, because it shows that there's this different set of assumptions that goes into the model, but you can model more or less the same kinds of things. So it's just a matter of picking your poison and seeing if it works. I don't know how did you try anything with tensegrities and CompuCell or anything approaching that, or what do you think?
No, he said that 3D CompuCell was difficult. And then they were working with someone to do 3D models. I could look up what they called it. And he thought that maybe my tensegrities might fit in with that. But I'd have to have learned a whole different method of doing tensegrity. And at that point, I didn't have time. So there are 3 or 4 different programs out there that I would actually like to try. Actually, there's 6 of them, if you want me to name them. One's called Open Foam. It's more of a for fluids program, but in a way, embryos are fluid.
Yeah.
So I don't know.
Okay.
There's lots of research one could do on different programs for looking at tensegrities in cells, and I don't know which one would work better, and maybe none of them would necessarily, because tensegrities are unstable, and really so are cells, but they're deterministic in that they form a sheet and stay a sheet even if there's wobbles in the middle, and it's not that your epithelial tissue is still intact. So yeah, it's sort of interesting.
You're right. Okay, one last feature before we go. So this is something that Hussein put in the Slack. And this is an artificial life software package called Vectorcosm. So it's kind of based on Boyd's, if you're familiar with the Boyd's paradigm, where you have these collectively behaving agents, and they have a YouTube video that I'm going to walk through screenshots of and then show at the end the GitHub repository. So this is a developmental update from May. This is called Vectorcosm, and so each of these agents, which are these little triangles, they actually have their own sort of neural control, their brain. And to do this, they're revisiting spiking neural networks. So this is at the level of kind of public understanding, so it's not highly technical, but they're basically using spiking neural networks. They're using event-driven spiking neural networks, so it only updates when it's needed. And so this reduces CPU time and scale as well. Then they have they can identify local wiring patterns that are similar to like connectomes or actual brains. The output adapters take samples of neuron spike times to provide meaningful numbers. So you have this output layer that takes things from the spiking neural network, as specifically neuron spike times, and provides meaningful numbers to the output. And so this is a real-time network mutation, so you can mutate parts of the network and get differential outputs. And this is a continuous wiring pattern, cellular wiring pattern. Now they say that networks are extremely efficient, and this just shows the entire data structure needed for a spiking neural network. So and the behavior that it produces. So this is, I don't know how many lines of code this is. This is pretty compact for something that you might expect to be not so compact. So this is an example of the environment here, where you see the agents moving around. Sometimes if there are no input signals, the agents don't exhibit any behaviors at all, and they just kind of sit in place, and that can be a problem because you want them to move around. And so if the network flatlines, the agent can't motivate itself, and so it can't act collectively, and it just kind of sits there. They actually have like this environment where the cells can, or the organisms can interact. They call them spike network organisms, and they do pretty well when working in contact with other spike network organisms, I guess. Spiking brains are not meeting the level of refinement of regular brains, so we get spiking brains, we get a lot of behaviors that are not like what we might see in regular organisms. And this is another screenshot of the environment. This is yet another screenshot of the network. And this is another shot of the environment. And so finally, this is the GitHub repository. Leavoya is the account, and then Vectorcosm is the repository. And they have a release in this repository. It doesn't look like they have a license. They have a README, which kind of goes over the basics of, the demo and the dev build. Then they have these instructions on how to clone the repository and build the program. And then you can use these different hotkeys to control the interface that we saw in the screenshots. And so Vectorcosm is an AI aquarium for digital life forms. It is a curious viewing experience and a science experiment in digital biology. Vectorcosm runs as a client-side JavaScript app in a web browser. It creates a world made entirely of SVGs, or scalable vector graphics. Starting Vectorcosm will create a tank with random life forms, or boids, plants and rocks. Boids will eat food, grow, reproduce, learn, and adapt, so they need to move around and do all these things. Vectorcosm also includes a suite of automated AI training simulators you can use to help boids learn basic life skills quickly. So they're just kind of highlighting the spiking network, but there are these AI training simulators that you can also use. So this is great work, and if you're interested, go to that GitHub repository and get involved in that. All right, well, I think that's all for today. All right, yeah, so thanks for attending, and I'll see you next week.
Okay.
Thanks.
Yeah.
Take care. Bye.
Bye.